Today we will be covering natural polymers. Learning objectives. Students will be able to fully understand the following topics by the end of the video. Proteins are a class of natural polymers which perform various tasks throughout our bodies. There are two different types of proteins, fibrous proteins and globular proteins. Fibrous proteins provide structural integrity and strength and are the main components of muscle, hair, and cartilage. Globular proteins are roughly spherical in shape and are the worker molecules of the body. Amino acids are the building blocks of all proteins. Condensation reactions between amino acids lengthen the chain to yield a polypeptide and eventually a protein. The basic building block of all proteins are the alpha amino acids. The general formula for an alpha amino acid is shown right here. The amino group, the NH2, is connected to the alpha carbon, shown here, along with the carboxyl group, right here, is also connected to the alpha carbon. The R right here represents a side chain, which can be any common substituent. These side chains can be either polar or nonpolar. Polar side chains have large numbers of nitrogen and oxygen atoms and are hydrophilic, meaning water-loving. Nonpolar side chains contain large numbers of hydrogen and carbon atoms and are hydrophobic, meaning water-fearing. Polar side chains are hydrophilic because they have dipole-dipole interactions with the water molecules. Solutions of amino acids are buffer solutions because of this equation shown here. What happens is that this side group right here is acidic while this group is basic. So when it dissolves in a solution, the hydrogen atom moves from here to this side, forming what is known as the Zwitter ion. Zwitter ion. And the effect that happens is when an acid is added to the solution, this side reacts with it, neutralizing the effect of the acid, and if a base is added, this side reacts with it, giving away an H plus proton to neutralize the effect of the base. Polypeptides are just a bunch of amino acids added together, which eventually, when you have enough of them added together, it forms a protein. And this is accomplished through a series of condensation reactions between amino acids. In these reactions, the OH and the H shown here between two different amino acids react to form water, and the remaining C and N, which need to bond to something, bond together, forming what is known as a peptide linkage or a peptide bond. Now this reaction can be continued by adding on another amino acid to the right, having it react with this OH part right here, and it would keep going on and on and on until you form a very large chain of amino acids, all connected by peptide bonds, which is a protein. The sequence of amino acids in the protein chain is known as the primary structure, which is represented by short three-letter codes for the amino acids. Looking at the example down below, we have lysine, alanine, and leucine in a row. So the shorthand primary structure of it would be right here, given by the first three letters of each amino acid. It is also understood that the amino group is on the left and the carboxyl group is on the right. The secondary structure of a protein is determined by the hydrogen bonding between lone pairs on an oxygen atom in one amino acid and a hydrogen atom attached to a nitrogen of another amino acid, and this will be made more clear on the next slide. There are two different ways these hydrogen bonds can occur. One is known as interchain hydrogen bonding, which ends up forming an alpha helical structure, seen here, because the hydrogen bonds occur between a hydrogen and an oxygen atom within the same chain. And if it occurs between two different chains, known as interchain hydrogen bonding, a pleated sheet arrangement occurs, which looks like this. Expanding on the last slide, we can look at the structure on the left and see that when the hydrogen bonds occur within the molecule, within the same chain, like here, 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 you can see that it forms a helical structure. And when it occurs between two different chains, protein chains, and it is normally almost a straight line because they're just lined up next to each other in order to form those bonds. And finally, the tertiary structure of a protein refers to the overall shape of the protein. It can either be long and narrow or globular, and the structure is maintained by intermolecular and intramolecular forces, which include ionic, hydrogen bonding, covalent, London dispersion, and dipole-dipole forces. Denaturation is the process of breaking down the three-dimensional structure of a protein. The 3D structure of a protein is crucial to its function, so when it is denatured, it can no longer perform the function as it is intended. Carbohydrates are another class of biologically important molecules. They serve as a food source for most organisms and as a structural material in plants. The most important carbohydrates are polymers made of monomers, called monosaccharides, or simple sugars. These monosaccharides are polyhydroxyketones and aldehydes, and the most important ones contain 5 carbon atoms, known as pentoses, or 6 carbon atoms, known as hexoses. A common hexose is the top right one up here, called fructose, and it is called a hexose because of the 6 carbon chain in the middle. Monosaccharides normally cyclize in aqueous solution, which means that they form a ring. In order to do this, they form the ring shape seen here, and then the hydrogen atom and oxygen atoms in this part will bond, and a bond will form between the oxygen atom and the carbon atom here, forming a new OH substituent group on the right, and a new oxygen-carbon bond right there. Polysaccharides are more complex carbohydrates formed by combining monosaccharides. A disaccharide is formed by the elimination of water to form a carbon-oxygen-carbon bond called a glycoside linkage. And large polysaccharides are formed when each ring forms 2 glycoside linkages. We can see this in action if we look over to the right, where we can see fructose and glucose added together to form sucrose. In this process, these parts of the glucose and fructose will react, forming a water molecule and leaving one oxygen behind to form this carbon-oxygen-carbon bond, called a glycoside linkage. On the bottom, we'll see the polysaccharide for cellulose, which is made up of individual glucose molecules, and each glucose molecule will end up forming 2 glycoside linkages with the other glucose molecules. Finally, we'll talk about starch, cellulose, and glycogen. All 3 of them are polymers of glucose, but they differ in the nature of the glycoside linkage, the amount of branching, and the molecular weight. Starch consists of 2 parts, amylose and amylopectin. These are both polymers of alpha glucose, but amylopectin is highly branched and has a much higher molecular weight. Branching occurs when a 3rd glycoside linkage attaches a branch to the main polymer chain. Starch is the carbohydrate reservoir in plants, restoring glucose as fuel for the plant. Cellulose is the major structural component of woody plants and natural fibers. Cellulose is a polymer of beta-glucose, so its glycoside linkages have a different orientation than in starch. Glycogen is the main carbohydrate reservoir in animals and has a similar structure to amylopectin but with more branching. Looking at the diagram on the right, you'll see that there are 2 different structures for cyclized glucose. One is alpha glucose and the other is beta glucose. If you look closely, you can see that they differ in these aspects because the hydrogen and hydroxyl group are flipped in each molecule.
Okay, so moving on to nucleic acids, where we find nucleotides, which are composed of 3 distinct parts. First being a 5-carbon sugar deoxyribose in DNA and ribose in RNA. Second part is a nitrogen containing organic base. And the third part is a phosphoric acid molecule, which is H3PO4. So now I'm just going to talk to you about DNA and RNA and basically just the ins and outs of both of these nucleic acids. So structurally, DNA and RNA are very similar except for three fundamental differences. The first one being that RNA is a single-stranded nucleic acid and DNA is a double. Second part is that, as we learned before, they both have different sugars. And then lastly, RNA nucleotides have a uracil base, while DNA has a thymine base. So the key to DNA's functioning is its double helix structure, as seen in this diagram. So as you can see, DNA consists of two twisted strands, and each of these strands has a backbone of alternating groups of sugar and phosphate. And these groups are really just bases, is adenine and thymine, and cytosine and guanine. So we know, obviously, that cytosine pairs with guanine. Sorry, let me circle that. And then adenine pairs with thymine. And we must know this because when these strands are unraveled, they serve as a template for attaching the bases back together. So we see during cell division that these strands unwind, and the new contemporary strands are constructed. And from these new contemporary strands, we can put them back together so that they are identical to the original one. So now moving on to protein synthesis, where we find a gene, and a gene contains a code for a specific protein. And these codes transmit the primary structure of the protein to the construction of the cell. And always remember that each protein or each amino acid in a protein has a specific code. And the code consists of a codon, which is 3 bases. So skipping over RNA really quick, we see this diagram of protein synthesis and kind of just like the basic steps of how protein occurs. Basically, RNA reads your DNA, then uses a code to construct the messenger RNA. And this molecule travels into a ribosome tRNA, as we can see, with an anticodon that hooks onto the mRNA, forming an amino acid according to the codons. And then these amino acids link together to form protein. So always remember that protein is always growing. And also a quick thing to remember is that protein synthesis occurs in cytoplasm. RNA acts as a messenger carrying instructions from DNA for controlling the synthesis of proteins. And the RNA molecules, which are found in the cytoplasm outside the nucleus, are much smaller than DNA polymers. And kind of just going back to DNA, we have learned that when you delete a whole base from the DNA double helix, you create a mutation. So what we have seen is that instead of deleting the base, you substitute the base for another one. And this simply does not really affect the DNA double helix at all. It just kind of is just a redundant process, but with no like distinct effect, as opposed to deleting the whole base, which would create a totally new mutation.